So antiviral drug resistance pathways allows us to develop a important insight into understanding cross-resistance and how to manage our patients that are failing therapy. So in this next slide, I've listed for you on the L-nucleoside pathway with the M204VI substitution, the sensitivity or cross-resistance profiles against all the other approved drugs. So obviously if you're resistant to an L-nucleoside with a 204 substitution, then you're cross-resistant to all the other L-nucleosides. But that isolate is still sensitive to adefovir, tenofovir, and because of the shared overlap with the entecavir mutations, the 180-204, then it's intermediate sensitivity for entecavir. Entecavir still has one substitution up its sleeve, so to speak. Let's now look at the acyclic phosphonate pathway with the N236T. Obviously the L-nucleoside analogues are active against N236T, but adefovir is resistant, and we have identified in vitro testing that tenofovir has intermediate sensitivity to the N236T. Entecavir is active to the N236T variant. The shared pathway with the RT-A181T substitution is resistant, cross-resistant with the L-nucleosides, cross-resistant with adefovir, shared intermediate sensitivity with tenofovir, but is still sensitive to entecavir. The naive entecavir resistant pathway, the 180, the 204 plus one of a 169, a 184, 202 or a 250, obviously resistant, cross-resistant to the L-nucleosides, cross-resistant to entecavir, but that isolate is sensitive to adefovir and/or tenofovir. For the multi-drug resistance, an example I've given there is the 181T, 233, 236, 250, this multi-drug resistant pathway. We recommend that you consult a reference laboratory and consider the role of interferon alpha because all 5 drugs are compromised by those particular mutational patterns. Now the clinical application of the power of the resistance pathways and cross-resistance profile is shown in the next set of slides and explains why the entecavir response is less than suboptimal in the setting of lamivudine resistance. So patients who are genotypically lamivudine resistant at baseline, when switched to their 1 milligram entecavir dose, you can see that the rate of resistance to entecavir is pretty brisk compared to the naive situation of less than 1% by 6 years. You see it's 6%, 15%, 36, 46, 51 and almost 60% by year 6. The same applies with the adefovir-tenofovir switch story. Patients who have failed lamivudine and/or adefovir and then switched across to tenofovir, which was the basis of the Van Bommel study, you can see that if you have a wild-type polymerase or a lamivudine-resistant polymerase at baseline, and then you switch to tenofovir, that the response is reasonable, almost as good as if you're a naive patient. But if there's a history or a documentation, I should say, of genetic evidence of adefovir resistance, this is shown by the orange plot, then 50% of patients are still viremic after 18 months. Now, when we look at these patients in more detail, depending on the genetic profile of the virus in those patients, can even further shape the response. If a 181 only, an RT-A181TV1 only is present, then you can see that the response is a gradual grind away, so that even by 4 years, 50% of patients are still viremic. And the response is really less than optimal compared to previous studies with tenofovir. The situation is even worse if there's an N236T present. And when the N236T is present, by 4 years, only 30% of patients are now under the limit of detection of virus, i.e. 70% of patients are still viremic. And we only see a response really within the first 12 months, where the viral load drops 1, 2 or 3 logs, and then from then on, the viral load is subsequently horizontal. And this is because the N236T is cross-resistant with tenofovir, and if you put the 181T in combination with the N236T, it looks like it's absolutely resistant in my view.